Hello again. I understand that I'm the last talk of the session, so I hope the day's been informative and productive, and I wish you all a safe trip home tomorrow. And thank you, again to Kevin and the SERG partners for allowing us to give you a recorded presentation of our work today. So this presentation is about our work on the biological control of emerald ash borer in Canada. We've been fortunate enough to have had SERG funding for a number of years to support this work. And while I'm presenting today, there's a large number of collaborators on this project. And whenever I do one of these talks, I like to start by acknowledging our partners. And I apologize that my bubble is floating over the Quebec flag. I'm sure Vero will give me a hard time later. So we have a number of partners within the federal government, within the United States, with industry. The folks at Bioforest have been very good about doing our release maps every year for us, the IC, and then a number of landowners across Eastern Canada that have allowed us to use their properties as release sites for biological control agents. And those folks, excuse me, without those folks, we probably couldn't do this work. And I noticed that I need to update my little graphic here because I'm missing some of our partners in the Maritimes who've been past couple of years have been giving us release sites. So I need to get those from Corey, I think. And also, so this is the team that's been part of the biological control program for a number of years. We've been going for a long time. So I introduced you to my team earlier. The folks that are working in my lab right now, the two fellows in the end, those are some of our graduate students who've been using the biological control system and the emerald ash borer rearing system at the Great Lakes Forestry Center for their graduate work, and that's been really interesting. I just threw some circles around the most recent members of the team. There, kind of the yellow circles. I hope they're showing up on the screen. Are folks in insect production in Quebec and then in Atlantic Canada. And we've actually been, it's all right, and I should say we've been doing this long enough that we've had folks retire out of the team, and so I just wanted to acknowledge those folks before I go on. So I'm sure people here might recognize Barry Lyons and Krista Ryle. They were frequent attendees of SERG. Gene Jones and Tim Ladd were our senior technicians on the project for a number of years. Gene ran this for a long time. And Tim took over for a little bit just before he retired. Rivera Lavallee was part of the program early on, and then I'm sure folks know John Sweeney, who is apparently retired. I teased him at a talk about 2 weeks ago, 3 weeks ago, about the number of emails that I get from John for somebody who is apparently retired. But again, this has been a long program, go back to about 2012, and lots of folks involved. And so I'm just the one who's fortunate enough to be able to present this to you today. So I'm going to talk about our program in Canada. This is folks who've seen this before. This won't be, this will just be a little bit of an update. The two questions we've had have been kind of, we've been doing the releases. Where have they established in Canada? That's kind of our proof of the pudding that we've actually done something. And are they dispersing? That's one of the research questions we've been looking at in my lab in the Sioux. So for the next couple of slides, I'm going to show you some maps of where we've released insects in Canada since the program began in 2012. And what this map shows, so these maps are kind of going to look the same, and then I'm going to focus in on the Maritimes because that's where we did our releases this year. And so what this map shows is the release locations of Spatheus gallinae, which is the one we began releasing in about 2017. So it's the most recent insect to join the team as it was. And it's one of the two larval parasitoids of emerald ash borer, and it's what attacks the insect as it feeds underneath the bark. So this is a zoom in to show the release locations in the Maritimes, and these have been coordinated by Corey Hughes, who I think is there, and his collaborators in the Maritimes. And the most recent sites that we did, it started in 2019, that Corey started in 2019, are there in the kind of pale yellow in Moncton and Cole Harbour. Near Halifax. So that's the releases there, but you'll see our releases since 2019 have been focused in the Maritimes, kind of following where emerald ash borer has been found. So we had some releases near Halifax, and then up at Sea Agate in 2019. The first releases in New Brunswick. And now you notice there's a gap for 2020 for kind of like obvious reasons. So this is the release map for Ubius sagrilli, which is the egg parasitoid of emerald ash borer. And just like to give you a sense of scale, that's an adult Ubius up in the corner there next to an EEB egg. So it's super tiny. Ubius has been released since 2015, and you might notice there's a few more sites in southern Ontario. So there's some sites. So we've been releasing this, all the way down, um, around the Guelph area, all the way up through eastern Ontario into Quebec, and then into the Maritimes. You notice there's some sites that we released Ubius where we didn't have Spatheus, and that's just because Ubius was approved. Spatheus was approved later than Ubius was, and so we just didn't. By the time we had approval to release that one, those sites especially in that kind of southwestern part of Ontario, were kind of too far gone to do releases. And so and by the way, these maps are just all colored by the year of release. So just there on the side of the screen. And again, here just showing up a blow-up map of our releases in the Maritimes. All these sites in the Maritimes have released both parasitoids. And what's kind of interesting here is that all of our sites in the Maritimes have also received all Canadian-grown parasitoids for Ubius and Tetrasticus, which is the one I'm going to show next. Spatheus is Spatheus we don't rear in the lab in Sault Ste. Marie, but Ubius and Tetrasticus we do, and all of those last two species released in the Maritimes have all come from our Canadian population. So that's kind of a little neat part of the program for the Maritimes. So finally, this is the map of release locations showing Tetrasticus planopenisi, and this is by far the most widely released biological control agent, emerald ash borer, in North America. Sorry, in Canada and in North America. It's been released here for I guess it's now 10 years. Next year will be 11 years. This year will be 11 years. And released all over in far south western Ontario. These are the first releases that have been done. Tetrasticus is also a larval parasitoid, so it attacks emerald ash borer when it's growing as a larva underneath the bark of the tree. The big difference between Tetrasticus and Spatheus is that Tetrasticus has a shorter ovipositor, so it tends to be able to do better attack in smaller trees with thinner bark. Spatheus has a bit longer ovipositor, so it does a little bit better in it does better in larger trees with about thicker bark, so it can handle that. And again, here's our maritime map, same as the other ones I just show you. All these sites have received, and what I want to emphasize here is that all of our sites in the Maritimes have received all three species. And the point of that is releasing all three species is to target the different stages of emerald ash borer, and also different emerald ash borer feeding in different tree sizes. So like I said, Tetrasticus is better as smaller trees, Spatheus does better in bigger trees, and then Ubius targets those egg stages. So you're trying to hit the population of emerald ash borer at different times, different size trees, and trying to reestablish some kind of control of those populations. And so the goal of the program has never been to end those outbreaks and prevent them from and sort of maintain those larger trees. The idea is to reestablish emerald ash borer in places where those larger trees that are hosting emerald ash borer are probably going to die, and then we can protect those smaller trees, those regenerating trees, as they come back on the landscape. And what I would also just emphasize here is that the pattern of releases has followed, the expanding range of emerald ash borer. So the insect has progressed further east. We've been making releases where there are large populations of ash that EAB can sustain. So when we do releases, we want to be able to make sure there's a bunch of emerald ash borer that are there that can that can sustain those emerald ash borer, those parasitoid populations, so they can establish and begin to spread. And by doing so, those larger trees will eventually die because they have so many emerald ash borer in them, but we're hoping to protect those smaller trees when they come back. So the question we get asked, and the initial goal of the biological control program, was to determine if these parasitoids could successfully establish in Canada and then eventually have an effect on our EB population. So the first question is, where have they established in Canada? And that's easy enough just to show with a map. So this is just our Tetrasticus release map, but it's the same for all the other parasitoids. In Quebec and Ontario, we have decent evidence of establishment of one or more species at just about every site in Ontario. And so that we know that the populations have been able to establish there. populations in the Maritimes, it's to be determined. The only we wait after we complete releases, so we do 2 years of releases, and then we wait for a year to let those populations try and establish before we come in and assess them. And the assessment, because the assessment involves removing some trees and then seeing what comes out of them in the lab. And so our population C. agus is just the first one that's old enough, been established long enough to be able for us to do those assessments. And so I think Corey was going to take care of that sometime before the spring, so we can have an idea of what's going on in the Maritimes. And then in a couple of years, we'll see what's going on at these other populations in the Maritimes. So the next question we get is, are they dispersing? I mean, it's well and good just to have the populations established, but if they're not moving around or they're not attacking other emerald ash borer populations, then they're not really doing anything. So that's a research question we've been addressing in my lab. So looking at dispersal, I'll just talk about 2 experiments we've done over the past couple of years. And this is just a photo of Megan Gray, who's the technician in our lab, and showing 2 of the methods that we use to assess dispersal on emerald ash borer. So at the top, that yellow bowl looking thing is a yellow pan trap. We attach this to a tree, you put some RV antifreeze in it, and the insects come along and they're attracted to the yellow and they blunder in and they die. And then you come along and you collect the fluid and you see what insects are in it. And then the thing that Megan is hanging from the tree is a new sampling device that the excuse me, that the Americans have come up with. It's basically a sentinel technique, where you establish emerald ash borer in these sticks of ash there, and then hang them out in the woods during the parasitoid flight season, and then see if they get attacked. And so we tried this for the first time. There was a paper recently published in the U.S., and we were interested to try it, so we gave it a kind of a trial run in the Algoma region last year, and I'll talk a bit more about that in a second. So we had 2 experiments. The first one, we were using yellow pan trapping, looking at dispersal from some of the oldest sites in southern Ontario. So in 2019, we went to 7 of these sites that had been established between 2014 and 2016, and we placed 12 trap locations around each site, and then we monitored them during the summer of 2019. That involved Megan and one of our summer students driving down pretty regular and going to all these traps and trying to see, and collecting these bowl samples. And then Megan spent most of COVID at home going through these samples in her kitchen. And so what we did is we had this sort of center, we centered these on each release site, and then we had a trap at 1 kilometer, 10, and 20 out sort of along each cardinal direction. And then we did the same used the same method in 2021, but we're doing a kind of a fine scale survey of the Algoma region. So we have 1 release site here at the very southern end of what's called St. Joseph Island. It's a Parks Canada site, and we were looking at trying to see if we could assess dispersal from this 1 site. So we established a pan trapping and an ash sampling grid, so we also sampled a couple ash trees across this region to evaluate spread away from that St. Joseph release site. So what I'm showing here is a map of those 2 studies. So on the right-hand side, we've got our 2019 study in Southern Ontario, and on the left, I've got the Algoma region study from 2021, and just an inset there showing where those are relative, in case you're not familiar with Ontario. And so all the black dots, all the dots are where we had traps. All the black dots, we didn't find any parasitoids, and the red dots are where we did, right? So if you look at our Southern Ontario sites, we had pretty good dispersal away from a lot of these sites. That's maybe not from some others, but not too bad. And then same thing here in Northern Ontario, decent dispersal, and certainly off this island. So what we did next was we wanted to see, what's the dispersal rate? How fast are they moving? So what I did is I calculated the time between when we did the experiment, when we were trapping, and the time since the first release at the closest release site, and then just regressed that against the distance between where the trap was to the closest release site. And when you do that, you wind up with kind of a regression like this. You get a positive relationship between I apologize, my bubble has landed on top of my own words. You basically get a positive relationship between the distance from the trap site and to the release site and the number of years since the first release. And if you take the slope of that line, you get the dispersal rate. And so we get a dispersal rate of about 2.5 or 3.5 kilometers a year for tetrasticus. This is all for tetrasticus. We didn't pick up Spathius in any of our plots. And this is consistent with field observations from some of Jean Douan's work, and also some flight mill studies that were done in Brian Occam's lab in Minnesota. So what this tells us is that the insects are dispersing at kind of the same rate or slightly faster than estimates from the United States, which is good. It tells us that they're behaving as they should, they're moving away, and that's kind of what we hope would be happening. So that's just from the yellow pan traps. When we did our 2023 Sentinel bolt study, so this is the other method we're talking about. So what we did, use those Sentinel bolts, and we hung them at 5 sites in the Algoma region. So here's our St. Joseph release site again. This is Sault Ste. Marie, and then we put traps at these 5 sites where we knew we had trapped parasitoids, so we knew that this was a positive test of the method. We knew we hoped we were gonna catch something if it was there. And so what we did is we had kind of a 4-week deployment of these traps in the trees, kind of in June and July. And what we would do is each of those sticks there would hang for 2 weeks. And then at the start of the second week, we would come and hang a new one. So during most of the summer, there were 10 of these hanging in a plot, and each of them out for 2 weeks. And what we do when we build them is those sticks are kind of sticking into a cup, and in that cup is full of rock wool and water, so to keep those ash sticks fresh. And these are all grown up in the EB colony at CFS. And brought them back to the lab, put them in the emergence cages to see if any parasitoids came out, and then when the parasitoids, when that kind of phase of emergence was done, we dissected them all to see what was inside. And so when we did that, and we plotted basically what we found. So the left-hand side here is the number of Tetrasticus planipennice larvae. So this is all for TET. That we collected from the bolts, and on the right-hand plot is the number of adults that emerged. And so, and then the bottom is the date that the traps were brought back in. So we started this kind of mid-July and then did it into August. So each 2-week period essentially. So we had 4 2-week periods. And what we see is we got each of the dots is just our sites. And then what we see is we definitely recovered both larvae and adults, which is what we're hoping. More larvae than adults, obviously. Oh, excuse me. But what we were seeing there is, excuse me, what we saw is basically the method works. It worked really well for collecting emerald ash borer, the parasitoids, and it certainly confirms that the insects are dispersing, that we're able to catch the Tetrasticus at these sites. And the nice thing about those traps, and I wish I could go back and show you the slide, but apparently you can't do that when you record your talk, is that it's a much simpler method to use, and a much simpler method to send out to people that we could have do sampling for us, so we could survey some of those sites in southwestern Ontario, and even into Quebec and into the Maritimes eventually, so we can have an idea of how those populations are moving. So it's a really cool technique, and I'm really kind of jazzed to try it. I don't think we're going to have a chance to do it this year, but starting in 2025, I'm interested in finding some places that we can use this method to really get a handle on what are these parasitoids doing, especially in southern Ontario, to control emerald ash borer, and if they're having an effect on these populations. So what's up next for the program? We're going to continue our release program in Eastern Canada. We have some sites, I think, penciled in maybe for next year in the Maritimes. I think we have 2 new sites in Quebec that Vero could talk about. I think one is with the Wabanaki near Quebec City, which sounds super interesting. Like I said, we really want to work with this dispersal range and impact of EAB. That's kind of the questions I'm really interested in now is, what are they actually doing now that we have, over 10 years of releases of these parasitoids in Canada. And then our final thing is we are really interested, I've talked about this a bit before at other SERG presentations and at Pest Forum, about sort of transitioning the way we run the program to one that's more led by cooperators, that we still are in the, that kind of transition was put on hold for a couple of reasons. But what we really want to get to is a place where our cooperators are taking responsibility for finding sites, getting permissions, collecting, receiving the agents from us and doing the releases and then doing those assessments if they're interested. And that's kind of the American model. That's what they do. We'd like to move towards that and so all that we're kind of doing in the CFS is those research questions that we're interested about Emerald Ash Borer, but also looking at, how do we optimize the program and then supporting the program by producing the parasitoids at the lab in Sault Ste. Marie and facilitating any imports from the States. That's kind of where we want to go. Hopefully maybe more about that in a year. But yeah, that's kind of the next phase of the program. So thank you very much for staying to the end and listening to this recorded talk. I appreciate it. If you have any questions, like I say, that's how you get a hold of me there. And I look forward, wish you all a safe trip home, and I hope to see you again next year at the SERG meeting. So have a great afternoon, and bye-bye.